on behalf of Cambridge Health Institute's Breakout COVID-19 series, Global Web Symposia, and our sponsor, Agena Bioscience, I'd like to welcome you to Responding to the COVID-19 Pandemic, Perspectives from Clinical Laboratories Performing SARS-CoV-2 Testing. My name is Nyla Ganatra, and I'm the host and technical director for today's event. Now I'd like to introduce our chairperson for today's webinar. He is Dr. Darryl Irwin, Vice President of Scientific Affairs at Agena Bioscience. Welcome, Dr. Irwin, and the microphone is yours.
Everybody, thank you, Nyla. It's my pleasure to be your chairperson today and also your second speaker. But first, I'd like to introduce to you Dr. Brent Zanke. Brent Zanke is a graduate from the University of Manitoba, where he did a medical degree, or completed a medical degree in 1983, as well as the University of Toronto with a PhD in 1993. Brent is a fellow of the Royal College of Physicians of Canada with a specialty certification in internal medicine and medical oncology. He conducted basic and translational laboratory-based research at the Ontario Cancer Institute before assuming administrative duties as director of the Cross Cancer Institute in Edmonton, one of Canada's 2 freestanding cancer hospitals. He was the provincial head of systemic therapy for Cancer Care Ontario from 2002 until 2006. Dr. Zanke also leads the multi-site Ontario Tumour Bioprofiling Initiative of the Ontario Cancer Research Network. This large-scale project is a part of an ongoing bioinformatics effort to organise clinical outcome measures with biologic profiles of human tumours. He also conducts large-scale biomarker and genetic predictors of colon cancer risk, having received funding from Genome Canada, the Canadian Cancer Society, and Ontario Ministry of Research and Innovation. Brent is the founder and chairman of ArcticDx, a private Canadian molecular diagnostics company, where he also fills the role of Chief Medical Officer. So thank you for joining us, Brent, and I look forward to your presentation.
Darryl, thank you so much. First, a sound check. I'm certainly hoping that everybody can hear me and hear the words that I have to say about our experience in using the Agena MassARRAY System in our recent efforts to convert from a molecular specialty laboratory into a high-throughput SARS-CoV-2 testing laboratory. And in the time that I have, what I wanted to do is emphasize a couple of things for people that maybe aren't familiar with the system, or those that are, how it could be modified to allow high throughput testing. What I want to do is emphasize a couple of things. First of all, the reliability, the remarkable ability to multiplex, and the ability to, in a very cost-effective fashion, process many samples simultaneously. I'm just going to say in advance that I'm a happy customer. I'm not receiving any compensation of any kind for this webinar, and so perhaps you can regard what I have to say as objective. So the laboratory that I run is located in Grand Rapids, Michigan. It has CLIA, CAP, and CLEP certification, and this is a little picture of home for us. It's in a beautiful recaptured industrial complex that we're very proud to call home. Okay, next. This is an outline of the processing schema that I'm going to just describe a little bit in each step. I'm going to use this as a template for organizing the talk to show you real images of what goes on in our lab, how we have addressed the issues of throughput requirement, but also, I think, quite importantly, how do we manage a large volume of potentially infectious biomaterial in a safe fashion. And so I'm going to just ask for the next slide that actually describes, I think, a really interesting tile feature of the work that we found ourselves in, that we, I guess half a year ago, were a specialty lab doing molecular genetic testing for rather uncommon diseases. But we have been recently pressed into service to process tests from nursing homes, from hospitals, even from a 1,000-employee egg farm, if you can believe it, and all kinds of agricultural workers in western Michigan and beyond. And so there's a very, very diverse collection of material that's coming that shows no signs of abating as the pandemic continues. Next. So I want to just talk a little bit about accessioning. The critical thing for us is that we recognize that for an enterprise that could be receiving 1,000, sometimes more samples a day, if there was a requirement to put all of the material into the LIMS system, it would dramatically slow down our ability to process samples and adhere to the 24-hour rule. And so, number one, we very early insisted that all material that is referred to us as opened and processed in a biocontainment hood. We don't have any opening of hazard bags outside of a biocontainment hood. And in order to accommodate the volume, we made a capital purchase for 4 or 5 new large 6-foot hoods, for which we're very proud of. So what you can see here are the nasopharyngeal tubes on the left-hand side that come directly from source. You can see the requisitions on the right-hand side. And what happens is that just two things occur. A barcode label is affixed to the paper requisition. It's matched to the tube, and then the requisition is scanned with a scanner that's located within the hood, and then the actual accessioning process of transmitting that information is done outside of the laboratory area. It can even be done by home. So this is an important feature that allows us very quickly to take the accession samples and move them on to the processing procedures that we have. We have about 10 or 12 people that work full-time in accessioning. We're actively pursuing an OCR type of technology where the scanned image can actually be converted into an accessioned record very quickly. The next step is that the viral transport medium tubes are then transported to another biocontainment hood where a team will take an aliquot of about 100 to 200 microliters of the VTM and mix it with lysis buffer and place it into deep well 96 plates that are made by Nunk that are currently a bit in short supply, but fortunately we've got a large amount of these on site. Next, the extraction process. We've quickly decided that the Agena EUA that probably people are familiar with that used Trizol was not really amenable to a high throughput environment. And we understand that there's an amendment to the EUA that describes omega magnetic beads as an extraction method. And so we've adopted this really as a LDT type of test where we're using the next generation omega beads, our beautiful Biomech FX robots. We've got 2 of these, and you can see the little post-magnetic platforms here. And I think probably many people on this call are familiar with the bead purification process and ultimate elution in a small volume of about 50 microliters of nuclease-free water. So this takes about an hour and a half. We can get to the point of the samples reaching the lab, the accessioning being partially done with the scan, the quick aliquoting of material into a 96-well format, and then immediately being processed on the machine. We're set up to do about 400 tests over the course of about an hour and a half. People that are familiar with the Agena system recognize that we then quickly proceed to the PCR SAP stage next. The way we do this is in a separate clean room using Agena reagents. We pre-pipette plates that are standard that are designed for the COVID COVID-19 program that we have. We order these in bulk and make about 30 plates at a time that are ready for use. Critical, this is a physically separated area to reduce the risk of any kind of errors or contamination. Then we go on to the PCR phase. We've got about 18 double-headed PCR machines that have the high capacity to be able to take these plates, put them on, and at various stages of completion, we can then immediately go on to the actual analysis. I want to just pause here and say that when we decided to set up, we were very concerned about capital expenditures and throughputs, and had briefly considered the qPCR approach that most laboratories are doing. And when we recognized that the qPCR requirements would actually be huge to get the output that we needed, we very readily recognized that on a single plate, we only need one well per sample, because all of the amplification segments could be analyzed at once using the resolution of the mass tag approach. So we wouldn't need, obviously, a separate well for one or two separate amplification products. And that we can do a twist positive control. This is the synthetic RNA, as well as a blank, but all of the other plates are actually the analytical samples. So the thing to emphasize is that on this particular plate, the number of samples that can be accommodated is actually pretty impressive. It's one sample per well, and if you have all of those PCR machines going at once, you can see that the throughput is actually quite impressive. For people that are unfamiliar with the twist synthetic RNA, you purchase it. It's relatively inexpensive. It goes a long way, and it has all of the amplification pieces that you need in order to prove that you've got positive PCR amplification within a plate. The other control that I think we'll probably hear more about later is the bacteriophage MS2 that's included early on in the RNA extraction process, that's included in each and every well. And this is a positive control for both the extraction and the PCR having occurred in every well of the system. The next process is the amplified product, or spotted on a spectrochip using a nanodispenser. We can show you a picture of this. This is our beautiful setup with two glorious MassARRAY machines. You can see an old-style nanodispenser on the left, the new one on the right, and a computer console that describes the output. An interesting sort of side piece for people that might have a little bit of a genetic epidemiology bent, I wanted just to tell you about some recent experience that we had that shows the diversity of the system. We serve various geographic locations within Western Michigan, and what we were noticing is that the pattern of positivities that were coming from different hospitals and institutions was a bit different. I don't want to burden people too much with data, but I just wanted to provide you a little bit of a simplified profile. What you have on the top is a little bit of a map of the genomic structure of the virus. And I have three patients here in each column, one a negative, one a positive from one site, and the other is a positive from a second site. And although there are 5 amplification segments that are included in the MassARRAY SARSCOP2 system, 3 nucleocapsid amplification pieces, as well as 2 open reading frame 1 and 1AB. I just wanted to show you something interesting about the 3 nucleocapsids. So the very first bit that you can see where the red arrows are, this is the column, shows you where you should see a mass signal that would be indicative of an amplification product. So this would be for N1, N2, and N3, which are segments of the nucleocapsid gene, which is on the far right. This is a positive from a local hospital, and you can see that the arrows are showing that we've got strong amplification and a clear signal. And the third column is from a nursing home where you can see something quite interesting that we're calling positives, but we're not seeing any amplification of the N3 product, and that every single of the positives that have come from this one nursing home have amplification of all of the SARS-CoV-2 pieces except for N3. And what we've noticed over the last couple of weeks, that the hospitals are now beginning to show small numbers of positives that lack this N3 amplification product. And we are in the process of sequencing the positives that we have just to try to understand why there's no amplification. And we suspect that what we have is a different strain, more than one in the community, that are showing different patterns of possibilities. And so we're quite interested in this, want to write it up as a potential genetic epidemiologic observation and just see where that particular strain might have originated. The part that I find most exciting is that nasopharyngeal swabs, which we've all had lots of, aren't very fun. In fact, if you've had one, you're not so keen on another. And perhaps if you're in a daycare or you're a university student or you are even a grade school attendee, you probably are just not so keen on this. And we're actively anticipating that there's going to be a broad need for testing in the educational system. So what we've done is over the last several while, we've been collecting paired nasopharyngeal and saliva collection pairs, in order to analyze them both using exactly the chemistry that I've described. And we have actually shown that there's terrific concordance between saliva and the nasopharyngeal viral transport medium. In fact, the amount of virus that's presented in saliva to the system is much greater than it is for the nasopharyngeal viral transport medium, because you're only sampling 25, maybe 30 microliters of nasopharyngeal material that then is diluted in 3 mils of transport medium, and then you take an aliquot of that and put it into the system. For the saliva, it's basically 1 mil of saliva, half a mil of preservative, and 200 microliters goes into this. And so we see that the sensitivity is quite high. In fact, we even see some positives in saliva that we don't see in the biotransport medium. The actual price of one of these collection tubes is about half what the nasopharyngeal swabs are, believe it or not. So we're in the process of completing a paired validation. The FDA requires 30 positives, and I think it's near perfect concordance. Maybe you can miss one. And that once that's validated, you can then start using this. And at the same time, your EOA will be evaluated. We want to convert all of our nursing homes and community testing to saliva, and we think that this will increase the throughput, it'll increase the acceptability, but most importantly, it really reduces the cost of collection, because instead of a registered nurse with full PPE, I can imagine a drive-through where you could hand somebody one of these things. You can see you spit into the little funnel. There are preservatives in the cap. When the cap closes, it presses on some tines that are embedded in the base. The liquid goes through, the funnel unscrews, a cute little cap goes on top. You invert it a few times, and then you're basically ready to go. And so we're very much pushing forward with this. We believe that we'll have it validated in the next short while, which will probably increase the demands for testing quite a bit. Grand Rapids is there in the little red balloon. You can just see sort of where we are in relation to Chicago, which is at the base of Lake Michigan, and Lansing, the capital of Michigan, there off to the right. So I'd be very happy to provide any kind of further information. I want to say that Agena has been exceptionally good to us in terms of troubleshooting, getting the system up and working in the lab, supplying expertise and reagents, and I'm very grateful to them. They've been terrific partners, and I just want to emphasize that Arctic and myself are not receiving any benefits associated with this webinar. So that's the conclusion of my presentation. I don't know whether you want to entertain questions now or whether we should answer the questions collectively at the end. But that's my 23 minutes, and I'm grateful for the chance to speak. Thank you.
Thank you, Dr. Zanke. The plan is we'll answer questions at the end, and I see there's a number of questions coming through. Thank you for submitting those. Keep submitting them, as we will have plenty of time to answer at the end of the presentation. So now I'd like to cover more of the technical details, but also the details of our validation and verification work as we were developing a SARS-CoV-2 test on the MassARRAY system, and how the test that we've developed, in addition to having some exceptional accuracy and sensitivity, also delivers low-cost and scalable testing. Now, one of the key linchpins in our fighting this pandemic is to offer testing to the masses, and this technology allows us to achieve that. All right, so today I'm going to cover the performance of the test and the concordance with orthogonal methods, sensitivity, limit of detection data. Following that, I'm going to cover some of the workflow and throughput capabilities of the Agena SARS-CoV-2 test on the MassARRAY system, and then lastly, look at the cost of this test and how we can deliver this in a low-cost, high-throughput format. So the key consumable with the MassARRAY system is our Spectrochip array, and what you see in front of you here is a 96-pad Spectrochip, and we also have 384-pad Spectrochips, having 4 times the number of features than the one you see in front of you. So each pad is generic to all of our chemistries, in fact. So whether you're doing methylation, whether you're doing genotyping, somatic mutation detection, or in what we're talking about today, infectious disease identification, you use the same Spectrochip. The pad binds to any DNA analyte mixture, and it enables detection of multiple variants on a single pad. In fact, we can genotype up to 50, for example, signal nucleotide polymorphisms or viral targets on a single one of those features. So that allows us to develop robust and redundant multiplex tests where we can have multiple assays which confirm detection of this of virus nucleic acid in this instance. And we're using upfront PCR and single base extension, endpoint PCR, such that we don't see any inter-assay interference. And because we're using mass spectrometry, we're detecting the nucleic acid molecule itself. So we are uninhibited by fluorescent crosstalk. The analyzer itself is a MALDI-TOF mass spectrometer. So MALDI-TOF stands for Matrix-Assisted Laser Desorption Ionization Time of Flight, which is quite a mouthful, so we shorten that to MALDI-TOF. And what's happening inside the mass spectrometer is on that pad is the matrix. So the matrix is a crystal that donates a proton, and we add a small amount of the amplified nucleic acid onto that pad. So we're co-crystallizing the nucleic acids with that matrix. And then when the laser pulses, the proton is transferred from the matrix over onto the nucleic acid, making it charged, and it moves into the gaseous environment. Now, that gaseous environment is actually a vacuum. The molecule is accelerated and it flies towards the detector at the top of the flight tube. The time of flight is directly dependent on the mass of the molecule, and the mass of the molecule is directly dependent on its sequence. And as such, therefore, we can determine the sequence of the nucleic acid fragment based on its time of flight. And what you have on the right-hand side here is an example of mass spectrum, where in the window that we're zoomed in on, we have two different viral targets here of the SARS-CoV-2. So these are two areas that we're amplifying that come out at different masses because they're different sequences. So you can see that those signals are very clear compared to the baseline, very, very clear peaks, which allows us to write automated analysis software such that you don't even need to look at these mass spectrums in the majority of instances. So the system itself delivers a number of features. Simple analysis software, as I said, which assesses all of the controls and the various different assays in the multiplex and gives you a detected, not detected, or an invalid interpreted result. Load and go chip processing. So at the front end, we have our thermocycling and amplification. And then once the amplification has finished, it goes into that blue tray that you see underneath the number 2 there. So the amplified plates go into that tray, and inside the instrument, the amplified PCR products are conditioned, transferred onto that spectra chip and analysed by the mass spectrometer without any further human intervention. Continuous access, door number 3 there allows you to access the reagents at any time, even while a run is processing, so that they can be topped up where reagents are starting to run low. And lastly, you don't need to wait around to take the plate off the instrument. We have analyte preservation, so a plate cooler which maintains a steady state temperature and preserves that sample if you wish to put it in as you go home and leave it on the instrument overnight and come back in the next morning. So a little bit about the SARS-CoV-2 panel performance. So just a little bit about the regulatory status that we are in at the moment. So part of the FDA's policy, they announced the capability for emergency use authorization. So under the policy for emergency use authorization, Agena advised the FDA that we completed our validation studies and we began shipping kits on June 23rd. And subsequently submitted the package to the FDA. Our intended use, and this is an abbreviated intended use, the SARS-CoV-2 panel for use on the MassARRAY system is a multiplexed RT-PCR MALDI-TOF test intended for the qualitative detection of nucleic acid from SARS-CoV-2 in upper and lower respiratory systems. Currently, it's an in vitro diagnostic pending FDA review, so we have not heard back from the FDA since we submitted our package on the 23rd of June. However, we have been assigned a lead reviewer and are in discussions with that lead reviewer at the moment. So this is something that's underway. But as with the emergency use authorization policy, once we've submitted our package, we're able to supply this test to the market, and we have been doing so for the last 50 or so days. So the Agena SARS-CoV-2 panel amplifies 5 viral targets. 3 of those targets are in the N gene, 1 target is in the ORF1AB region, and 1 target is in the ORF1. And in addition to that, we have a control where an external MS2 template is spiked into the sample. That MS2 is then co-extracted with the viral molecules amplified and detected to confirm that as a procedural control. So having multiple targets for the viral RNA really provides some advantages. First of all, it increases accuracy. Having multiple targets moves you further away from the issues of sampling error when samples are very low copy number. The redundancy also allows us to be resilient to mutations underneath primer sites, in that we have 5 viral targets, and our test, as I'll show you shortly, we only require 2 of those targets to amplify to call the sample as a positive. So if there were to be mutations underneath our primer positions in one of those targets, it wouldn't then push that result into a questionable result or a false negative. And that MS2 control there is there to monitor all of the extraction, the amplification efficiency, detection, and reagent integrity. So mutations in the SARS-CoV-2 have been of concern of late. There was a paper published just recently in very late July about a mutation at position 26,340 that was published in the Journal of Clinical Microbiology that was reported to cause failure of some assays that were on the market, particularly those that used qPCR or QRT-PCR, because there were only 2 targets in that test, and a mutation underneath the primer caused one of those targets to fail and therefore had an ambiguous result. This SARS-CoV-2 virus does not seem to be a rapidly mutating virus, but of course it is a virus, and therefore there is quite a number of SNPs and mutations that are occurring and have been characterized in the genome. So this is a very real risk. So the MassARRAY offering those 5 targets, comparing that to some other technologies on the market. Now, most real-time PCR tests are using either 2 or 3 viral targets, and as I mentioned, a mutation underneath one of those target regions would cause the result to be ambiguous and questionable, in that you have 1 positive and 1 negative as the result of a 2-plex RT-PCR. So with the MassARRAY, we're able to put all 5 targets into that one single reaction and increase that robustness to any mutations that can occur. And the other thing that was brought into question with that publication is whether the SNP at that position did truly cause an issue. And that's because the primer sequences for the technologies that were in question there were not available to the users. So for transparency, Agena's assays are all open source. All of our customers have access to the sequences of our primers, such that they can do ongoing surveillance against public sequence databases of the SARS-CoV-2 genome. Now, not that we're pushing that responsibility onto our customers, we're also doing that surveillance ourselves as well. So let's move into our limited detection study. So here, as a limited detection, we looked at bowel samples or bronchoalveolar lavage and also nasopharyngeal samples. So with limited detection, the FDA is quite prescriptive as to how these studies need to be performed. So we start with a serial dilution. So here we have a 1:2 dilution of control viral material spiked into negative sample matrix. So a 1:2 dilution where 3 technical replicates is done at each dilution point until and you're looking at where you get less than 3 of 3. So basically the concentration of viral material where it drops to less than 100% detection. On the left-hand side at the BAL samples, it was 0.2 copies per microliter. On the right-hand side for the MP samples, it was 0.4. And then using that crude limit of detection, we then continue on to do 20 replicates in a finer range or finer dilution of viral material. And that confirmed that, yes, our bowel samples were at 0.3 copies, where all 20 replicates were positive. And the MP samples were 0.4 copies per microliter, where 19 of 20 samples were positive. So quite a low limit of detection, which is afforded by that multiplexing of 5 viral targets in our single reaction. So then moving on to the clinical evaluation. Here we have done a clinical evaluation of both bowel samples as well as nasopharyngeal. Now, the FDA has again been prescriptive as to what is required here. So at the top with bowel samples, because getting sufficient numbers of positive BAL samples is a challenge, or was a challenge at the time and still is, we were able to do a clinical evaluation study where it was spiked into negative clinical samples. and we had 30 of the clinical samples where there were 0 copies or it was negative, and 100% concordance there. And then spiking in 0.6 copies per microliter, we had 20 of 20 coming up as positive. So good concordance with our limit of detection. So the clinical study being performed at a 2x of the limit of detection that was determined in the previous LOD study. We also ran 4x there of 5 of 5. And then we did run a 6x at 2.4 copies per microliter, where 1 of 5 was not detected. On further investigation of that false negative here, which is at 6 times the limit of detection, that sample was retested at both a 2-fold and a 4-fold dilution, and both results were detected. And we concluded that this was indicative to inhibition due to insufficient alcohol removal at the last wash step of the RNA extraction. And we added to our instructions for use additional steps to ensure that alcohol had been or is sufficiently removed to circumvent this happening again. Then in the nasopharyngeal samples, we received samples that had previously been detected with a comparator test, and of the negative samples with the comparator test, we detected the negative samples that we received, we actually detected a significant number of negative samples that were detected as positive, and this was quite concerning because we saw a negative predictive agreement of 72%. So although the positives were largely in agreement, so 41 out of 42 positives were detected, and I'm sorry, I've just noticed, and the reason I'm slowing down here, I've just noticed we've got the columns on this table at the bottom the wrong way around. Anyway, what it results in is that we have a positive predictive agreement of 97%, and then we have a negative predictive agreement of 72%. Now, 72% negative predictive agreement is certainly not something that we were very happy with at the time, so we needed to do some further adjudication as to why 16 of those samples that were determined to be negative were coming up positive in the Agena test. So the comparator that was used there was the Hologic Panther Fusion SARSCOP2 assay. Now, it had a published limit of detection of 0.01, tissue copy infectious disease of 50 per mil. And of course, the Agena had a 0.4 copies per mil. So when we converted TCID50 through to copies per microliter, the Agena did have a much lower limit of detection. So it was probable that many of these samples were actually positive. But they were very low-level positive because they were collected from patients that were symptomatic during the pandemic. Because previously we had run a series of negative BAL samples where the BAL samples had all been collected prior to the pandemic, so prior to this year, and they all came up as negative. So we hadn't determined a false positive issue. So we then employed the Bio-Rad digital PCR test. And that Bio-Rad digital PCR test for SARS-CoV-2, again, is an EUA panel with an advertised limit of detection of 0.6 copies per microliter, which is closer to that of the Agena panel. And then what we observed is out of the 16 negatives that were detected as positive on the Agena panel, 15 of them were detected as positive on the Bio-Rad assay as well. And also the one that was not detected on the Agena panel was reported as positive. So the one other discordance in the previous study was also not detected as positive on the Bio-Rad digital PCR. So all up, 15 of the 16 false positives that we observed in the previous study and one false negative were resolved in favor of the Agena SARS-CoV-2 assay by that adjudication with digital PCR. So now taking that into account, our performance is a limit of detection of 0.4 viral copies per microliter with 100% positive percent agreement with the adjudicated results, and a 98% negative percentage agreement, again with those adjudicated results. All right, so the MassARRAY system offers scalable system throughput. So the up front, of course, as Brent explained, you need to be able to do your accessioning of your samples and your RNA extraction. We do have experts on automation on hand that we can help you with that RNA extraction capacity. But then once we move after the RNA extraction into the Agena kit, which is the RT-PCR, the SAP extension, loading it onto the MassARRAY, and then the data analysis, what this system offers is completely automated data analysis and load and walk away in the mass spectrometer. So once you've amplified your product, you load it onto the mass spectrometer, and that does the sample conditioning and analysis, and then an automated report. And down the bottom here are some examples of possible outcomes from the automated report. So here you can see it's a very simple CSV format, so that it's compatible with transport over into your LIMS system, identifying the sample name, the location, the QC status, whether that's a pass, a warning, or a fail. If it's not a pass, messages as to what has been observed, and then the sample status there. So as you can see, that top sample has QC passed, SC2 has been detected in that sample. The next sample is QC passed with SC2 not detected. In fact, that's the next two. And then we have some examples of where there are warnings in place. Sample number 5 there has a QC warning status because one or more of those QC markers has failed, so that is the MS2 endogenous control that was added in has not been detected. However, SC2 has been detected, and part of the decision tree is that result can be reported because SC2 was detected in that sample. But if both the controls as well as SC2 are not detected, then the sample is reported as invalid and recommended for reaccessioning. So the instrument itself is loadable in a continuous format. We do have 2 different instrument capacities. One is a 384 instrument, and that's the one that I'm looking here. So in a 384 instrument, the instrument can be loaded at the start of the day, and it starts processing. And then once the sample has started processing and has moved over onto the mass spectrometer side, which is around about 2.5 hours later, after the first 2 384 well chips are loaded, then another 2 plates can be loaded, and then continually 2 plates being loaded every approximately 2.5 to 3 hours. So if you were working in 24-hour format, that does mean that you're able to do 6,144 samples. But if you're only operating a standard 8-hour working day, it would mean that you can load the first 2 plates, second, third, and the last 2 plates, so the 4th set of 2 plates at the end of your working day at hour number 8. Those plates continue to process through the night. So a standard 8-hour working day would allow you to process 4 times by 768. So I believe that's just over 3,000 samples in a standard 8-hour working day. Likewise, the lower throughput 96 well instrument, again, that continuous loading of plates over time. So your first 2 plates going in, and then approximately 2 hours later, another 2 going in. So in a 24-hour format, allows you to process just over 2,000 samples in a day, and in a standard 8-hour day allows you to process 1, 2, 3, 4, 5 times by 192, so just over 1,000 samples in a standard 8-hour working day. And lastly, on supply and cost because constrained supply has been a significant issue, and of course, cost is always an issue for the health system and for testing laboratories. So first of all, we've been very transparent on what the cost of this panel is. So clinical laboratories can process more than 6,000 samples per day, and this is a quote from our CEO. And our test is priced at under $10 per sample. So it's generally in the vicinity of $10 per, well, it is at $10 per sample is the cost per test. Higher volumes can get us slightly under $10 per sample. So this sort of pricing allows significant scalability and large-scale testing being accessible to the massive population. And now with reliable supply, what you have learned so far in this seminar is that we don't use fluorescently labeled oligonucleotides. So our reagents come from unique supply chains that we are not constrained by the volume of testing that's happening on other technologies. And also we've worked to secure and source and stock all of our hardware as well as our consumables such that they are ready for immediate deployment. We are not on allocation at this point in time, nor do we plan to be on allocation, and as we're equipped to supply millions of tests every month. So lastly, the Agena SARSCOP2 test is a high-throughput, low-cost assay for the qualitative detection of the SARS-CoV-2 virus in upper and lower respiratory specimens. It allows accurate detection because we have 5 multiplexed assays in addition to our control assay across the SARS-CoV-2 genome. Scalable throughput, allowing you to process from just small numbers through to thousands of samples per day with a single instrument. Low-cost reagents with automated result reporting saves your lab both resources as well as dollars. And we have that reliable supply, meaning that we don't have shortages and we are not on allocation. All right, so thank you for attending. We will now move to answer some questions. So I'm going to quickly try and scan which questions have come in. So first question is a question from Denisha Wallach. So Brent, what lysis protocol do you currently use for RNA extraction?
We use a protocol that is pretty much supplied by Omega in Atlanta. We did a little bit of a modification based upon some experience that Agena provided us with regarding the addition of the MS2 RNA target, but it's basically right off the shelf from Omega.
Okay, great. And another question about your accessioning here. What, if any, robot do you use in the hood to initially aliquot the specimen into the lysis buffer?
None. That would actually be a terrific addition to the whole process. I guess what we would like to be able to do is take a 96-well array of the 3 mil viral transport medium and have that automatically pipetted into the deep well nunc plates, but we don't have that, and that would be something worth doing. We're a little bit constrained by the amount of space that exists in the hood, though. But that's an excellent thought. That's something we should look into.
Okay. Well, that question was from Jeremy Hart. So, Jeremy, if you have any capacity in your laboratory and would like to get in contact with Brent, please get in contact with us.
We'd love to hear what you're using.
Yeah. All right, thanks. So the next question is for myself. So does the MassARRAY System, a new machine, chipret module, does the deck allow for 4 94-well plates or 1 384-well plate? Okay, so we have we have 2 instruments. We have a 384-well instrument and a 96-well instrument. Both instruments take 2 plates at a time. The 384-well instrument takes 2 384-well plates, so 768 samples at a time, whereas the 96-well instrument takes 2 96-well plates. So it's not a case of the 384 takes 4 96, it takes 384-well plates. So your robotics and your thermocyclers need to be set up for 384 format plates with the 384 system to achieve that higher throughput.
Can I just add a note onto that? We very much would like to use the 384 well plates, but in the extraction protocol with the requirement for 2 mils of capacity per well, I don't think that it's possible to get a 384 well plate that has that capacity that could then go on our particular robot. And so we would be in a position where we would have to then pipette the 96 wells into 384 well plates before it went into the PCR, which we thought had too much potential for error. And I'd be interested to know whether anybody can do the RNA extraction chemistry using a robotic system in the 384 well plates. I've no doubt that the back-end PCR spectral chip, fly process can accommodate 384. It's just the amplification that's a problem. Not the amplification, the extraction.
The extraction, yeah. Well, I'll put that question out to our audience here, and if anyone does have experience with doing RNA extraction in 384 well format, would you mind chatting, putting your response into the chat, and we'll put you in contact with Dr. Zanke. So our next question is for you, Brent, from Juan Carlos. I was just wondering, why weren't there any peaks associated with the ORF-AB and ORF1 target displayed in the graph? So I presume that a spectra is contained in Dr. Zanke's presentation. Let me just switch to that slide for you, Brent, because I have control. I put the slide here.
I understand the question. I just didn't include them in that image because it was too cluttered. I just wanted to use the 3 nucleocapsid amplification segments just to illustrate the point. But for the positives, Darryl, you might have mentioned this, that the calling convention is that if you have one amplified product, it doesn't consider it to be positive. You need 2.
Correct. Yeah.
So what we found in those positives is that we had pretty much across the board amplification except for the N3. And if it was just one sample, we would have thought that this was curious. But the fact that out of that particular nursing home, every single sample misses this N3, and it's unique until quite recently, where we see that same pattern diffusing out into the community. And that the positives that came from that started, we think, in the kitchen, where 5 out of 5 kitchen workers were all positive with that same N3 deleted version.
Mm.
I mean, it doesn't really, from the point of view of getting in the test, processing them, and reporting them, it doesn't really mean anything. But we just thought that it was actually a very curious pattern of seeing these particular viruses move through the community that we could distinguish.
All right, and we did, we have had a couple of responses regarding the 384 well extraction. Most of those responses are that, you can extract in 96 well format and then use 4 96 well plates to move into the 384 as you're doing. And that's commonly what all of our 384 well users are doing at present.
Yeah, that's right.
All right, so I've got another question here. So what is the output of the MassARRAY? Does it require manual analysis? So no, the output is an automated report. I'm just moving back to it now. So this is the automated report here. So the data is displayed, the spectra and everything else that you've seen throughout the presentation is displayed, but then from this type, what we call the typist software, you initiate a report. So it does an analysis of that raw data and then comes out with this report at the end of the day, which is a comma-separated values file with the details of the software that was utilized, the time of the report, the samples, their locations on the chip, which particular run and chip or plate was run, and the location being the well position. And then the QC status and the sample status, so whether the QC markers have passed, warned, or failed, and then the actual interpretation of the sample status being detected, not detected, or invalid if it was not detected, as well as no control assay detected in the sample. So it doesn't require any manual analysis. The raw data is there for you to review, but in most instances, you don't want to be looking through 384 well spectra, so it's an automated report. And I think we've got time for just one more question before we reach the top of the hour. Brent, are you currently experiencing reagents shortages? How is this impacting your laboratory?
We're actually not. No Agena reagents. It's the NUNC 96 well plate that's a problem, that I think they're not even accepting orders until like December, which is a little bit sad. We've been able to source out the biomech tips, which I think is just specific to our particular machine. And I don't know how broadly those 2-mil round bottom NUNC plates are used. But other than that, there's no shortage whatsoever.
All right. Well, I'd like to thank you, Brent Zanke, for your contribution to today's webinar. I hope everyone online has enjoyed the information. There are a lot of questions here. So anybody whose question we did not get to, we will be contacting with an answer offline via your details. We'll get in contact with you via email to answer your questions. And thank you for the volume of questions that have come through to today's presentation.
And just one last word. We relied on a lot of mentoring and help getting this very complex system up and running in almost no time at all. And I'd be very, very happy to talk to anybody that's contemplating the same thing and give them some help and advice and guidance.
Great, thank you, Brent. And I'm sure people can Google you and find your details, but if not, just come through to Agena and we'll.
Yeah, I'm there. Google me and you'll find me.
All right, we'll hand it back to you, Nella.
Wonderful. Well, I'd like to thank our presenters for today, Dr. Zanke, Dr. Irwin, and of course, Agena Bioscience for sponsoring today's webinar. And finally, I'd like to just thank all of you for attending. We sincerely appreciate your willingness to share your time with us. Stay safe and have a wonderful rest of your day. Thanks so much. Goodbye, and we will end the webinar now.